Hello, everyone. Hope you're doing well. So what I'd like to do in this video is walk through ideas that appear on the multiple choice section of the Unit 6 test. So let's begin with repressible and inducible operons. A repressible operon is normally switched on but can be repressed or switched off when appropriate. An example of this is called the TERP operon, which is the tryptophan operon. Normally, this operon is switched on and it produces enzymes that help produce tryptophan, an amino acid. So when an operon is switched on, the operator site is open, what we see here, and then RNA polymerase can attach to the promoter, which is in green, and then zip along and transcribe the genes. Now, this happens because, again, bacteria constantly need tryptophan in order to make polypeptides and proteins. Now, if you eat a meal and feed tryptophan to the E. coli, then what that tryptophan will do is attach to the repressor and change its shape and activate the repressor. When the repressor is activated, it will dock on the operator, and that blocks RNA polymerase and prevents transcription. So you repress or switch off the transcription of those genes. Then when the tryptophan becomes incorporated into polypeptides and tryptophan levels in the cell drop, then the repressor switches back to its inactive form, the operator is cleared, and transcription begins again. So again, that is an example of a repressible operon. Let's see here. It looks like we have an inducible operon. An inducible operon is normally switched off, but can be induced or forced on. Good example of this is the LAC operon. These are the enzymes that are needed to the genes that are needed to produce enzymes that will create lactose and help break down milk sugar. Now, normally, this operon is switched off. So the repressor is active, it docks on the operator, and again, that blocks RNA polymerase, which makes sense because E. coli normally don't need to make enzymes to break down lactose because E. coli don't make lactose. But if you consume a meal that has dairy in it, some of that lactose or milk sugar can dock on the repressor, alter its shape, and inactivate that. When you change the shape of the repressor, it no longer docks on the operator. RNA polymerase can transcribe the genes. You make the enzymes that break down lactose, and then lactose levels in the cell will fall. When lactose levels in the cell fall, the repressor goes back to being active, docks on the operator, and prevents transcription. So that's inducible and repressible operons. Let's see. Next, we're going to look at liver cells and lens cells and their activation in eukaryotic cells. Well, I guess liver and lens cells are only going to be in eukaryotes. But looking at this, the idea is both lens cells and liver cells, cells in the human body, they all have the same DNA, right? All these cells have instructions for making a copy of a person. But what differs are these little activator proteins. So in a lens cell, certain activator proteins will be present. What they'll do is dock on specific enhancer regions on the DNA outside the gene. And then that helps situate these transcription factors in purple here. And then transcription factors are needed to situate RNA polymerase. So RNA polymerase, the sort of white teardrop structure, it's going to transcribe the genes. But RNA polymerase needs to sort of be positioned. So these transcription factors act like a booster seat or a car seat and position the RNA polymerase. But the transcription factors have to attach to something, and they attach to these activator proteins. So again, different cells are going to have different activator proteins present. Those different activator proteins will grab the DNA in different locations or different enhancer regions and cause the transcription of different genes producing different proteins, so the cells specialize in different ways and do different jobs. So that's discussing that. Control of gene expression in eukaryotic cells. Let me pull something up here really quickly. Sorry about this. I didn't have this prepared. But what I'd like to do is look at a lecture from chapter 18. There we go. Regulation of gene expression. So prokaryotes, we know that since transcription and translation basically happen simultaneously, you got to control gene expression in prokaryotes through operons. So we have your inducible and repressible operons. Eukaryotes, we have a nucleus, so we can separate out the events of transcription and translation, so it gives more points of control. So this is all in the Chapter 18 lecture, and this particular illustration or figure is found in your textbook. All right, so let's look at this. First point of control is access to the DNA. If you add methyl groups to DNA, the DNA coils up. If the DNA coils up, you don't have access to the code, the gene is shut off. If you acetylate DNA, if you add these acetyl groups to the histone tails, the DNA uncoils, you get access to the code and you can transcribe the DNA. So our first point of control is whether or not the code is even accessible. Next thing we're going to look at are events that can happen prior to transcription. And this is what we talked about earlier. If certain activator proteins are present, it's going to grab DNA in different locations, and then you'll transcribe different genes. Now, what happens once those genes are transcribed? Well, after transcription, there's other stuff we do. One thing we do is we add the 5' cap in the poly A tail. The 5' cap helps situate the messenger RNA on the ribosome, and that poly A tail slows the digestion of the messenger RNA. Remember, mRNA is basically a disposable code. It's a disposable copy of information. And so enzymes in the cytoplasm chew that up, and the poly A tail slows that. But one other interesting thing that happens is after DNA has been transcribed into pre-mRNA, we cut out sections of the pre-mRNA. Those sections are called introns. And then the remaining segments, called exons, get stitched together. I just remember introns stay in the nucleus, exons exit the nucleus and get expressed. Now, what this can do is lead to what's called alternative RNA splicing. So when you cut out the introns and stitch together the exons, these numbered segments here, the combination of exons can actually vary. So you can use the same DNA code, but get slightly different polypeptides. Again, this is a low stakes situation because mRNA, it's disposable. If it doesn't work out, get rid of it, make different ones, right? The DNA is where things are at. Next, this is, what happens on the way to the ribosome. Messenger RNA can get captured. So here in red, this is like your mature messenger RNA. It's left the nucleus and it's moving toward ribosomes. Well, we have little snippets of mRNA called microRNAs. And if those microRNAs have sequences that resemble any sort of, if they find mRNA that's at all reminiscent of information from viruses, it can actually be captured and destroyed before translation can occur. So microRNAs can silence genes. Okay, so again, you can produce a messenger RNA, you can edit it down and create a mature messenger RNA, but even before it gets translated, it can get captured and destroyed. And this is, again, it's called RNAi or RNA interference. Let's see. And even after translation occurs, you can tag proteins and then they're basically recycled. Okay, so all these are ways of controlling expression of genes in eukaryotic cells. All right, let's look at restriction enzymes. Restriction enzymes cut DNA, but they cut DNA at very specific sequences. We know that enzymes are really particular, right? So here would be this EcoR1 enzyme, and it will cut the DNA along a very particular sequence. And the cool thing about this is when it cuts the DNA along a certain sequence, it doesn't cut directly across. It makes a staggered cut that leaves what are called sticky ends. If you recall, these bases, they're going to be able to hydrogen bond with other other molecules. So what's amazing is if you can use DNA from two different sources and cut them with the same restriction enzymes, they'll have complementary sticky ends. And since all life uses the same code, ATCG, if you cut samples from different species with the same restriction enzymes, they'll still have similar overhangs or complementary overhangs, so you can actually merge DNA between different species. So step one is to cut with restriction enzymes and create these sticky ends. And then you can recombine DNA from two different sources. And then you use another enzyme called ligase to stitch together the sugar phosphate backbone, and then you have a completed molecule. Let's see, how to insert a gene of interest into a bacterial cell. Okay. Inspiration, glowing bacteria. Let's see what we can find for this. Sorry, let me clear out some of this stuff. So, sorry, I'm just sort of making this up as I go, trying to find images that would be useful. This is relevant for the lab you're going to be doing next week. So the basic idea is you could take a gene of interest and you cut it with a restriction enzyme. You could take a bacterial plasmid, which is like a little vector or vehicle for transmitting genes, and also cut it with the same restriction enzyme. Then you can recombine the DNA from the two sources, use ligase to stitch them together. And then because bacteria are really good at taking up exogenous DNA, you can actually get that gene for making a fluorescent protein into bacterial cells, and bacteria will produce that green fluorescent protein. So this is similar to what you'll see in lab. You'll be able to make some bacteria that have a plasmid that produces a protein that glows under UV light. Okay. Let's look up gel electrophoresis. Let's say explained. See if we can find some images for that. Okay. This is sort of nice. So when you create this agarose gel, within the gel are little air bubbles and some polysaccharides, some big sugars. And you load your samples, your DNA samples, into these wells. Now, DNA has that sugar phosphate backbone, so it has a negative charge. And then when you run current through the gel, the negatively charged DNA is going to be attracted to the positively charged end of the gel. Now, because different DNA fragments have different lengths or different base pair sizes, different base pair numbers, the DNA is going to separate out according to the size of the molecules. Bigger molecules are going to get caught up in the gel and they'll move more slowly, so they'll stay closer to the wells. And then smaller fragments are going to move farther down the gel. Okay. Let's see, reading gels. Well, here you can use the idea of an uncut gene and then a digested gene. If you have a piece of DNA that hasn't been digested or exposed to a restriction enzyme, it's in one piece and then it will run out to a certain distance. But then if you use a restriction enzyme to cut the DNA into, say, two pieces, then those smaller pieces will travel farther down the gel, so you can create these different banding patterns. Another idea related to this, if I could pull things up here. Again, I appreciate your patience as I sort of fumble around with all this. Another idea here is that if mutations occur in the DNA and you don't cut the DNA, you can again visualize that on a gel. So let me move down to later in the lecture here when we get into electrophoresis. Oh, this is sort of helpful. Here you see the EcoR1. Here you can move DNA from two different sources and stitch them together with ligase. And then you move the plasmid into a new cell. So that's very nice. Okay. So here's getting into PCR. Again, you load the DNA in the wells. It's attracted to the positive end. Excuse my dogs, please. I apologize for that. All right. So looking here is a gene for hemoglobin, and the restriction enzyme DD1 can cut it in 1, 2, 3, 4 places. So you create 1, 2, 3 fragments, one larger and 2 smaller. So you run it through a gel. Here's your larger fragment. Here are your 2 smaller fragments. But if you have a mutation in the DNA, you change the order of the bases, one of the restriction sites may be altered and the restriction enzyme doesn't cut it. If it doesn't cut it, well, then you just have 2 larger pieces that are left. So you can recognize that change in the banding pattern of the gel, electrophoresis gel. All right, the Hershey-Chase experiment. Let's look at the Hershey-Chase experiment. This is the one with the blender. Here we go. So what they did in the Hershey-Chase experiment is they wanted to find out what is the molecule of transformation, what gets passed on from virus to bacteria to change the bacteria. And so they grew 2 batches of virus, one in radioactive sulfur and the other in radioactive phosphorus. The idea is sulfur incorporates into the disulfide bridges of proteins. So if you grow some of the virus in radioactive sulfur, you're labeling the protein code. Then they grew another batch of virus in radioactive phosphorus, and phosphorus incorporates into the sugar phosphate backbone, so you label the DNA. So they let the viruses infect the bacteria with whatever they passed on. Again, they didn't know if it was DNA or proteins. So they let the viruses infect the bacteria, then they separated them with a blender, boom, boom. Then they put the fluid in a centrifuge to spin that down. The heavier DNA forms pellets down the bottom of the test tube, and then the smaller viruses stay in the supernatant. And what they found is that the supernatant had radioactive sulfur, showing that the protein coat stayed outside the bacterial cells. But then the pellet glowed with radioactive phosphorus, so it's DNA that gets passed on. So DNA is the molecule of heredity. Next, we're going to look at the idea of semi-conservative replication in DNA. The idea there is that when you copy DNA, one of the strands is original and the other serves as a template. So here you have the parental strands in pink, and then the daughter strands are in blue. Now, again, they determined this with sort of an ingenious experiment. It's the Meselson and Stahl experiment. Let's see if we have a good explanation for this. So in the Meselson Stahl experiment, they had 2 versions of an isotope that was incorporated into DNA. One used a heavier nitrogen and one used a lighter nitrogen. So DNA growing the lighter nitrogen, well, it's going to have a lower density, so it's going to be higher in the test tube. DNA that's grown in the heavy nitrogen has heavier strands, so it's lower in the test tube. Now, what happened is they were able to replicate the DNA through one round, and what they found is that you have this Goldilocks effect, that the DNA that's allowed to replicate for one strand is right in the middle. So you had one heavy strand and one light strand. So that shows that after one round of replication, you have one original strand and one new strand. And so it grows in the middle. So that's the idea of semi-conservative replication, that if you have DNA that grows in these 2 different isotopes or replicates in these 2 different isotopes, then that creates different weighted DNA molecules. And then if you allow replication to occur, one round of replication to occur, then you're going to get something in the middle there. So that's the semi-conservative model. DNA replication at the replication fork. So things to remember here. The leading strand grows in one piece going toward, that's not great. The leading strand grows in one piece growing toward the replication fork, that 5' to 3' direction. And then the lagging strand grows in the opposite direction. It's still going 5 to 3, but it grows in the opposite direction, and it's in those fragments, those Okazaki fragments. So just make sure that, leading strand is in one piece, lagging strand is discontinuous in those fragments. And then, of course, ligase can stitch that stuff together. Let's see, components of an operon. We've already sort of been through that, right? You have the operator, the on-off switch, the promoter. It's a lovely evening, and so people are out walking their dogs, and my dog's going crazy. Let's see. I'm sorry about that. Hey guys, please don't be like that, please. Okay, so the idea is there's a redundancy in the genetic code, so you can have different codons, but it still codes for the same amino acid. So that allows for some point mutations. You can substitute one base for another. But if it's in like the second or particularly third base in a codon, you may still get the same amino acid. Another thing to remember, there are a few stop codons. Stop codons don't actually code for amino acids. They code for that release factor that releases the polypeptide. So it's not an amino acid. It's simply a message saying, stop making this polypeptide. RNAi or RNA interference. We've already sort of talked about that. We've talked about activators, transcription factors, and gene expression in eukaryotes. We talked about restriction enzymes and restriction sites. Again, if there's a mutation in the restriction site, then you don't cut the gene. If you don't cut the gene, then that changes the banding pattern in the gel. Types of gene mutations. Let's see here. There are some different types. If you recall in your packet on that protein synthesis paper lab, there's like a missense mutation, nonsense mutation, insertion, deletion, stuff like that. That's all good stuff to review. So a missense mutation just means you're substituting one base for another. Could it be bad? Maybe. Could it be neutral? Maybe. Could it be helpful? Maybe. Nonsense mutations, that means you get an early stop codon. That's always bad. Insertions or deletions, again, change your reading frame, so those tend to be worse than anything else. Well, I guess it's in chapter 17. If you want to go review that business, you can sort of look at stuff on mutations, and that should give you some review there. But review this stuff, review the FRQs, and that should take you quite a ways for the test. If you have questions, let me know. Good luck with studying. I hope you do well, and I'll see you on test day.